Okay, it looks like most people have come in from the waiting room, so I'll get started. Hello, everyone. As always, thanks for attending. This will be our February session of the Funk Biogeography seminar series. Today we've got Pablo from the University of Miami. Some announcements first. So I think I mentioned this last time as well. Keep an eye out for the inaugural issue of our IBS newsletter. So that'll be coming up very soon. This will be like nice once a month email to get some news on society happenings, seminars, events, some highlights on frontiers articles that have been recently published, and hopefully a lot more as it develops. So keep an eye out for how to get that newsletter once it's released. And of course, our preview for next month, we have Catherine Graham from the Swiss Federal Institute for Forest, Snow, and Landscape Research. That'll be on March 25th, and there will be news coming up soon on the time for that and the contents of the seminar. So keep an eye out for that as well. As always, the way that this works is about a 45-minute talk and 15 minutes or so at the end for questions. So if you have general comments, concerns, things that don't need to be read out, you can use the chat function on the left side. Or if you'd like to submit a question, you can click that Q&A button on the right-hand side of your bar. That'll go into a text box, and then we will read that out on your behalf. And as always, thank you so much for attending again. If you weren't able to come today or you would like to view this again later, this will be on our YouTube channel at International Biogeography. If you want to keep an eye on what we're doing, possibly get involved in the society, I'll direct you to our website at biogeography.org. And if you want to check out our journal specifically, Frontiers of Biogeography, the link for that is biogeography.pensoft.net. So with that, I will turn it over to Marta to introduce our speaker.
Thank you very much, Hikaru. Welcome, everyone, to the February Funk seminar. It's great to have you again. My name is Marta, and I am the Vice President for Public Affairs and Communications at the International Biogeography Society. And today I'm pleased to introduce our speaker, Dr. Juan Pablo Quimbayo. Dr. Quimbayo is an assistant professor in the Department of Biology at the University of Miami in Florida. He has a PhD from the Universidade Federal de Santa Catarina in Brazil. He conducted his postdoctoral work at several institutions, including the Smithsonian Environmental Research Center, Sao Paulo University, and the Ohio State University. Dr. Quimbayo's research seeks to understand how ecological and evolutionary processes shape marine biodiversity across both temporal and spatial scales. Working at the intersection of community ecology, macroecology, and biogeography, he and his lab focus on reef fish assemblages to uncover the mechanisms structuring biodiversity from local habitats to broad biogeographical regions. Using trait-based framework, spatial analysis, and large-scale datasets, Dr. Quimbayo and his team investigate how environmental gradients, historical processes, and species interactions drive community assembly and functional diversity. His work integrates theory and application, generating insights that advance our understanding of biodiversity patterns while informing conservation and management strategies in our rapidly changing oceans. So today we hear from him about how he uses trait-based framework to understand marine biodiversity, and we could not be more excited. So without further ado, I turn it over to you, Juan Pablo. You may share your slides now.
Perfect. Thank you, Marta, for the introduction, and Hikaru for the invitation. It is my pleasure to stay here, start to put a little salt water in the Biogeography Society, and this is great. And today, yeah, you're right, Marta. I wanted to start talking about a little how can to use a trait-based framework to try to understand the marine habitats in general. Let me only try to turn my screen here, and I think this is working correctly.
It looks great, thanks. Yeah.
Okay.
Go ahead.
Okay, thank you again for the invitation. It is my pleasure to stay here. And today, again, we started talking about how use the trait-based framework to try to understand the marine biodiversity in general. When it started talking about biodiversity, we come in two different images for the mind. The first imagine coming is, for example, the tropical forest, when you have the Amazon, there's a lot of animals there. But at the same time, you have in the marine habitats the same type of the habitat. You have one habitat has a lot of biodiversity, and this is the coral reef. The coral reefs in general can include different animals, and for example, more than thousands and thousands of animals using the same habitat to try to survive there and living there. The coral reefs is very important by habitat to try to maintain the biodiversity because it help us to understand what happened in general, but in the same time help to in a different ecosystem service to understand what happened there. But to try to understand the biodiversity, the people using different metrics and to try to understand these different metrics, in the first moment, 20 or 30 years ago, the people start using the taxonomy of diversity to try to understand what happened there. And with the time and the evolution of the knowledge, many people start to move for another different metrics to try to synthesize the organisms and understand the organisms has available. One metric, for example, the people start to collect a lot of DNA information and start to you understand a little more about the phylogenetic diversity in general. And using the trace to people try to understand what is the organism state there. And this is the type of the things. In the last year, I started working more and to try to apply all this biodiversity index of all use of the species traits to try to understand what happened in the marine habitats. What is important to understand the functional diversity of all the traits is you have this class imagine for cat bender and barres and collaborators basically say, hey, when you have the traits here, you can start to understand the communities in general. And all these processes is related with the evolution of phylogenetic historical process. But in general, all is basically this, the species trait can provide insights about the ecological process. You understand the communities in general. But at the same time, this use of the trace can to apply in different spatial and temporal scale. For example, he started thinking about how can apply in different from the local, regional, global is here. You can start to understand ecological interactions. You can apply environmental filters, for example, dispersal and biogeography process to try to understand how change the community across the space and the time. In one example, when I try to see, you understand about the cleaning interactions, for example, between, you can see this is a big shark coming, swimming here, and this small fish try to swim very fast to arrive there. And during this fast interactional between both species, it's possible to try to extract a couple traits to try to understand what is the influence the traits for this interaction. For example, you see this shark is big animal, but this is small fish is a small animal and basically try to remove ectoparasite there and can transform this interaction in one method to try to understand why the ecological interactions is important to understand the communities. In other words, I transform my metric in what is the organisms to do in the system. But at the same time, we can't use another type of the traits to try to understand, for example, the dispersion. For many different animals, include fish, mammals, birds, is the body size is very important to try to understand what far one animal can arrive in one specific place. And the people start using these specific traits to try to understand the movement between the species. For example, today is possible to understand during this moment many different species. For example, the Brazilian coast start to move for the Africa coast, and use is very related with the body size, for example. And this is important to try to understand the traits, to try to you understand what happened in the future in this movement between the species. But at the same time, the traits is sometimes reflect all the evolutionary process the organism has. For example, the reason one organism has one specific shape or one specific mount position is determined of what is the type of the diet the organism is using. And all this evolution process but can using the one simple approximation to try to understand what happened there. And in other words, the trace is basically one simplification of the ecological process to try to understand how interacting the process in different scales. But the question is, what are the traits? How I can use the trait? And the first thing, what are the traits? Well, the trait is basically one morphological, physiological characteristic with the organism has to the individual two organisms. And this specific simplification can to help to apply in all different scales I try to describe here. For example, if see this is more, this is fish, this parrot fish here for the Brazilian coast, you can to see this organism have one specific size. For example, this medium size has one herbivore diet and it's diurnal species. It's basically I transform my species or my number of the species in one specific characteristic of the organism to try to understand a little more about what is the organism is there. But when I compare it with another organism, for the Brazilian coast, I can see it is different. Now I have one small species, one invertebrate species, but both have the diurnal characteristics. In other words, when you start to compile many different traits, you can start to understand what happened. And this is the first study we do to try to compare more than 26 traits for reef fish communities across the Atlantic and the Eastern Tropical Pacific to try to understand how is this process can interact between different scales. And part of the advantage to using traits, for example, is very related with application across these different systems and using different animals cross tags comparison to try to using the traits because can to simplify it in only one metric. Can to link organism respond. Many organisms can to change your reproduction to respond of the climatic change, for example, or can use it for experimental approach when it's limited and basically try to understand the relevance of the functional ecosystem service. But when combining all these traits, can to start to understand or calculate a couple of metrics, and many different people start using these metrics to try to complement all the taxonomy metrics that people use in classic in a couple of years ago. For example, in the first moment, I wanted to combine all the different traits. For example, in this table here, I have my rows, I have all my different species, and I have in the columns all the different traits. For example, in the first column, I have the body size. In the second column, I have the diet. In the third column, I have the habitat. But when I combine all these different combinations, I can decide one functional entities. I can repeat the same exercise several times with all the species I have and use this information to try to calculate my first metric. For example, try to calculate my functional richness, basically to try to calculate the distance between the species or how the species is dispersed in one space. In other words, I transform my traits in numerical number to try to understand what is important to understand. But what is important, you understand the functional diversity or functional richness in this case. Many different things related with functional richness can to reveal how environmental filters, for example, the insulation can influence the community ensemble. Let's go to thinking about one island. What happened with this one specific island? When one organism arrived there, it need one specific characteristic to arrive there. And obviously this limitation in the insulation can predict when I start using, for example, the traits to try to understand what happened there. But we can use another type of the metrics, for example, the functional evidence. Can to start using the functional evidence to try to understand the ecological role. What is the reason I have one species in the middle? And to try to using this metric, I can combine another important thing, another metric, for example, abundance, to try to weight how the organism is in the space. When I have high values, for example, I can say, hey, my functional evidence is more very distributed. This is important to understand the change in the communities, because sometimes to see when you lost one species taking more time to detect change in the abundance, for example. And this is important to try to use in this metric. Or when you have a low functional evidence, you can expect all the species are all representing only one part of the species. In other words, I reduce all the species just in this part here. Another metric you can use is the functional divergence. Is basically to try to understand what is the limit or the spectrum I have in related with my functional index here. In other words, all the species in this part here in the borders species is more different, have this different specialists or strategies to maintain in this space. And this is important because in this case, you can detect rare species, but when you have all these species around here, you can say, hey, this my species is more generalist. You understand how change my community for the specialist community for one more generalist community in general. To remember how can apply the species traits I mentioned in a couple of slides ago, the importance to understand about how cross system. And this work for Catalina Pimiento and Public Science with collaborators she basically combined different taxes to try to understand how is the functional space in this case. She combined mammals, she combined sharks, she combined fish, invertebrates, turtles, reptiles to try to put all in the same level to say what happened. And she found in this study basically mammals have the more 20% of the functional space is occupied for mammals in general. And this is the reason mammals is so important in general for this study here to show how is the different the trait this animal has there. But at the same time, we can use this specific approach to understand what is happening in biogeographic terms. For example, in this study, she showed, for example, in the North Pacific, in just in this part here, is totally different. 50% of this functional space is occupied in this part here. When you compare, for example, the South, it's totally different. In other words, I can use my functional traits to understand how the organism is biogeographic organized in general. We can predict or can you understand how the organic can to respond, for example, for different environmental effects. For example, in this study for Conlan and collaborators, he proposed in the first moment is many different fish communities will start to reduce the body size according the pressure or the temperature. I mentioned in the first moment, many organisms can to change their reproduction season or can to change how is the moment to growing to try to maintain and respond to environmental factors. And this is one good example to show the fish can to move from the big size to a small size. And sometimes you can see the same things in terms of the composition of the trophy level or the trophy gill composition. Basically, you can expect that you have uh, high predators, and in the end, you can see more low levels of the trophy levels in general. When he basically, when he tried to probe this response, he's basically have the temperature hitting this axis and have the uh, body size here, and basically he's across Australia, he detected the community in general start to decrease the side level according when the temperature is starting to increase. And what is this? This is important to try to understand what happened in one climatic scenario in the future when the temperature in the ocean starts to increasing and increasing more and more and more. And the traits can to help us to understand what is the consequence related with environmental effects in general. But the traits can to help us to relate the ecosystem functions. For example, when you finally you understand what is the organism do in one is ecosystem, you can expect it to calculate, for example, how is the bioreduction there, how much the biotis fish can remove there, and how is the predation, and start to understand all these cycles that can to help us to understand the ecosystem function. This is the more challenging part, for example, in marine science, to try to connect how one specific trait can to move for this specific ecosystem functions. But we working in try to renovate, innovate in this part and try to uh, more exact measures related with the ecosystem functions in general. Today, after for this big introduction, I really wanted to focus in three different examples. I wanted to start talking about from interaction to community and how can to use this trait-based approach to understand what as the communities happen. After I would move from local and global scale, and finally I will present the two examples related with from the past and the future, and how can use a specific trait-based approach to see human impacts, for example. In the first moment, I wanted to travel. I wanted you to travel with me to try to understand interactions in the ocean. In the ocean, it's possible to see the more common interaction can to see in the ocean is the clean interaction. Basically, one small fish or one small shrimp coming from another animal and try to remove ectoparasites. And this relationship or this interaction happen very common. It's very common to see in every place in the world. It's possible to see. But this interaction is only possible because a couple organisms try to explore opportunistic, and this is the case when you have facultative species or dedicated species. Basically, these dedicated species developed a specific strategy to swimming behavior and one specific characteristics to do the best service. In another words, to try to remove ectoparasites. And many people say this fish is so important to maintain the coral reefs or the community healthy. When I start to my career, I start to understand this type of interactions to try to see, hey, what happened when I have my network connectivity, my network here. I have in this column all my cleaners in one specific island and all my clients here, and I start to see what happened. And for my was surprise for me to see in this resource, all the facultative species has more abundance in general but have more connections. It's expected you can more dedicated species has more connections because it's more exclusively to do these specific interactions, but this is not happen. Basically, dedicated the faculty species can to clean more to try to understand a little more and to start to understand in global scale, I start to expand my studies to compile the different data for different publications and observations. And for me, was a little surprise to observe that, for example, the dedicated species, the species only cleaning all the time or clean all your life is more concentrated in a couple places in the world. For example, it's more in this part of the Atlantic and is in the Indo-Pacific Ocean. But in the Africa coast and the Mediterranean coast, these species are never arising. Why? What is the first question coming in my mind to try to respond? And many is related with the moving of the how is the evolution change or how is the host diversity change across the time. As you probably remember, many in different moments the plate start to move in across the time, and all the biodiversity hotspot, the marine side, the marine habitats start to move across the ocean, and this is the reason now is here. And this is what's the first question: why I have this pattern? And when I try to investigate, obviously this is very related, is I have one basically all my dedicated species, or my cleaner species is more concentrated in the tropics and is more concentrated in one specific longitude. But when I start to see the how change in terms of the species richness, again, is more concentrated here in the Pacific, but in the terms of the proportion, it changes a little. As you can see here in the Atlantic, for example, has more proportion of the species, the cleaner species. But what happened in terms of traits? And this was the next question to try to see, hey, when I see the specific traits, I can predict more dedicated species is very specific in your interactions, but have in different places in the world, maybe have different combination of the traits. But when I start to explore these type of things and you understand what it really happened in terms of the dedicated species in the functional space, these organisms occupy, basically I can continue to detect one small functional space. In other words, independent of the place or the marine province, this organism is very specialized or very rare, functional rare in general, and very restricted. In other words, I have dedicated species is very functional rare and restricted in general in comparison for the faculty species. This is one way to try to apply it trade-based approach to understand what happened in biogeography terms. But after to understand what the traits can to influence, I continue to investigate how can affect my interaction, how can affect the relationship between my clients and my cleaners. And I start to see it in general when you have the dedicated species very limited in terms of the traits, is the only reason I can find nested network. In other words, basically dedicated species can to clean in general. And this was different than the results I found the first moment in one island. This is very related because the place this organism happen is only developed more one body size and living in groups. In other words, this is the reason I can expect it or can explain my nested network in general. And this was very surprised because initially was unexpected to a small fish can to determine the network analysis I can find in this general group. We try to expand this trait-based approach. We start to investigate different interactions for fish. For example, my colleague and me compiled for the last 30 years, all the studies was published in the Brazilian code related with interactions and try to understand the nestiness and the modularity there. And we found in general, when you understand about nestiness in general, the trait explain better all these nestiness is related more generally species, basically more generally species or has the opportunity to explore different type of the diet can to help to maintain the nestiness in one big network. And this was a little expected because many organisms can to explore different habitats, basically move between the network and the different interactions. And this is the reason can expect this type of the results. But was surprised to understand what is the reason when I see the modularity, low trophy levels contribute more for my modularity. In other words, this is a little opposite to the first results. And we continue to investigate to try to explain what happened here and what the trait can explain. I try to explore another different trait, for example, the body size, how the body size can to determine my different parts I can observe in this moment. In general, we can use the trait-based approach to understand the interaction of the species and move between local scales to regional scale to global scales. Now, in this line to understand what happened in general, I wanted to move from the local to global scale and understand what is can to provide here. Well, the first thing, in general, I use more on the water visual sensor. This is one destructive technique to try to understand what the community has there. And usually one diver coming here using one tape measure to measure all the one specific area and all the animals in one specific area is a count. In the last 50, 40 years was collecting a lot of information in the south of the Brazil, and this was the paper was published in Time Fish, which was renovated the last year with more data to try to understand what happened there. And the first moment, now I wanna you and me move to Brazil. And in the Brazil, we have one specific place here is the Santa Catarina state, more in the south. And this state is very particularly important for us, the marine people, because it's one transition zone. In other words, it's the moment the tropical water coming for the north and subtropical water uh, come to meeting together. In other words, I can expect in my fish community, we combine it between subtropical species and tropical species. And another important thing related with this specific location is the last limited of the distribution of the reef in the South Atlantic. And this is so important because we start to investigate all the islands in this region and all these islands here in green with marine protected area and all these islands in the south is not protected area, but you can see here the difference between the temperature here. For example, in red is more tropical island temperatures and more in the south is more subtropical. We start to combine all these years and all the traits to try to mapping what happened in terms of the trait, how the trait composition changed in the time. And my PhD student published this first paper to show how is the community basically changed in the time. From the first year and the last year for this initial comparison, she basically using 40 different traits. She classified in different feeding, growing, and surviving attributes. And she described here in general, my community changed across the time. For more details, basically, the organisms start to coming for one moment when fusiforms, living impaired, large animals, high levels of the trophy levels for one community more reduced. In other words, more solitary species, more sedentary species, and more small species. In other words, she started to note one change in the composition traits. And this was surprise because in theory this has all this moving oceanography moving all the time and you have a couple places protected there, but we can see this general pattern for the North Islands and the South. We say, okay, we see one change in the composition traits and how can to you understand what is the composition trait will affect my community. And we start to investigate the same pattern there, but now change the question and try to understand how is my composition change across the time. And to try to do this one approach, we use and continue to use a base approach, a trait-based approach, but this time we're working in try to use in beta diversity metrics. Again, we have here in the north all the tropical islands and the south more the subtropical islands. And we combine all different traits we explained before. We have here the reproduction, growing, and survival traits to try to build in, again, complex whole or functional space to try to understand what happened. And finally, we use beta diversity metrics to try to describe why they changed there. And the first moment when I see the species richness there in the time, all the soft islands in the blue start to have low values in comparison with the islands in the north. This the tendency is more negative in comparison for these islands in the north. But when I start to see in terms of the species ratings again, in more in taxonomic way to try to compare after for the functional way. We see it in general for the species even if it's more constant and is opposite weight in terms for the species ring. You can see the islands in the south is more in the bottom, and the islands in the south is more in the top. But the trait composition we following the same pattern we observed for the taxonomic way, basically was no surprise in general. But however, when I start to compare the beta diversity to using the first year for the second year and the third year, and all the time using the first year to compare for the next year, we expected the species composition change because in the first study she showed the trait composition change. We can expect the same pattern here, but for surprise, we don't observe this time the change in general. For the North Island, you see the pattern is very constant, and this was maintained in terms of the abundance. And when compared with another in the South, was the same patterns. In other words, we don't see any change in the species composition. I don't can see any homogenization because in general, the people expect when you reduce the functional traits or you reduce, for example, the body size or all this diet, it's expected the community is more homogenized. In this case, we don't found this pattern in this location there. Maybe in the future, we need to continue to investigate what I can expect in the future to try to understand this pattern we continued or not. Now, I would like to move for one regional scale. I wanted to describe here the Eastern Tropical Pacific. The point I wanted to include the Eastern Tropical Pacific is because in many different global assessments, the Eastern Tropical Pacific is not included, and many cases because the people think don't have enough data. But I will show today has a lot of data available, ready to collaborate and to do anything related with the Eastern Tropical Pacific. For example, and the Eastern Tropical Pacific is so important because it's one region is basically insulated from another. Is taking all these arrows here can see it. For the south, the Peruvian current is one of willing to limit to quote all the tropical species and subtropical species, and this happening the same for the California current. Basically, you have two different currents limit the distribution of the species from the north and the south. 3,000 millions of years ago, the Panama Isthmus divide the Eastern Tropical Pacific to the Caribbean, and for the another part of the Pacific Ocean is basically more than 5,000 kilometers the distance. In other words, this is very insulated region, and probably many people in this talk hear about the Niño and La Niña. These climatic anomalies is very strong in this part in the equator. For example, in 98 has very strong Niño and affect all the corals in Galapagos, and many corals there disappear in one specific place. A couple of places protected, continue to see corals there, but all these climatic anomalies affect very strong this region. In one general terms, the people start to include the tropical Eastern Pacific, and this is first analysis, the functional approach for Muyo and collaborators. He showed here basically the Eastern Tropical Pacific in comparison with another region has basically one small number of the species in this first bar, and the number of the combination of the functional entities is low, but when you see the functional space, is very similar with another. In other words, it has one species there, has the similar traits, can find another place in the world. In one general collaboration with people from different countries, we put together more than 3,000 underwater visual sensors with different areas, different samples, and different samplings for. And we're using this data to try to combine it with the traits to try to describe how is the functional diversity change across this emblematic region. First was very challenging to try to put together because you see here it has a lot of different sampling area and was the first challenger to put up together. And we using, for example, Bootstrap to control this variation in the samples in general to try to compare. But the first results showed when finally mapping the species richness change across the Eastern tropical Pacific, again here in Costa Rica, has a lot of species there in comparison to this region here in Colombia coast and a little here in the south of the Mexico, has this all the species richness. But when you start to explore the functional richness, controlling for the species richness, we only found a couple places here in the south of the Mexico. Is very different in terms of functions. In other words, in general, the Eastern tropical Pacific don't have many different combinations of the traits there. However, when I start to explore the functional evidence, we start to detect it high values just in this part here. And this is important because this region is very, has a lot of endemic species around this region here. And we try to use another functional metrics to functional dispersion, basically how it is dispersed for the centroided. And we found a more different relationships across the Eastern tropical Pacific. Can detect a couple places, it has more high values around, but this is very related with the community hubs there. What is most relevant for this study was to found in general, we increase the functional evenness or has functional evenness, positive functional evenness when increase the gravity. And the gravity was one metric was published a couple years ago. The people try to understand how is the effect for the people for the coral reef and basically calculate the distance between the coral reef and the human populations. And in this study, we show here the gravity has one positive effect. Basically, when you have more people, you can see more better distribution of the functional evenness. But maybe the reason found this pattern is because we remove all the different animals have different traits. For example, we remove top predators, we remove herbivorous big or large herbivorous fish there, and this is start to homogenize my functional space there, and probably this is the reason I have the results here. And this was for the functional dispersion is basically when you reduce the area available, is the species is more near around the centroid, and this is the reason I have this effect here. Now, I wanted to move for another example here. In this case, I wanted to discuss a little about the mesophotic reef. This is a little challenge in different ways because you have for the 0 to 30 meters, you have the shallow reef, and all this, the coral reef people investigate a lot of related with the shallow reef in general, but we start to investigate more between the 30 and more than 90 meters. If you thinking about, hey, maybe one submarine can go there, but in general, when the people found a lot of studies in the shallow and very deep, all this middle is basically the people start to investigate now because has a more tools to try to do more investigation here. The first results Rocha and collaborators found here is all the species richness or all the composition of the species richness is totally different for the species in the shallow. And this was important because in the past, the people start to propose we can expect that many shallow species use the mesophotic reef one refugia, and this not necessarily is true because have different composition of species here related with this shallow. In other words, and to try to translate these in terrestrial organisms, the same expectations in terms of the altitude gradient, but in the positive way. We basically Russia showed this specific pattern to say the shallow reef is totally different in terms of the beta diversity, and we start to investigate more about this specific mesophotic reef and try to see the community is totally different there and can to do, and it's only possible to investigate with the specific technical diving, and this request a lot of training, a lot of effort to try to understand what happened there. But this is important because the people start to say, hey, this is the refuge, but this is not totally true. We see here, which when compared, for example, in terms of the species richness in the shallow, we see it here in the Pacific Ocean, all the species richness is very high in comparison for the species richness around the Atlantic. But when it start to go more deep, we can see this pattern change a little. The difference is no more high in comparison for this shallow reef. And finally, when arriving in the 60 and 90 meters, you can see the species or the proportion of the species richness is very similar. And this happened in local scale and regional scale. But when you start to explore in terms of the trade, what happened there, we start to see change in terms of the families. For example, many families or their herbivorous fishes start to reduce in general your abundant or species richness because in mesophotic reef, the temperature, the life of start to decrease. In other words, the algae due photosynthesis there is not necessarily available. And basically, this is part of the reason the species composition is start to change and the trait composition change. We see, for example, many herbivorous species start to decrease, and in the same time, many carnivorous species in yellow here, the gropers start to increase, and another family, the butterfly fish, start to increase when increase the depth. But this happened in the same time with the diet between the organisms here. Can to observe this different pattern there. But what happened in functional terms, basically with, again, combining all these different traits to try to understand what is the different, and obviously it's very clear the shallow have here functional redundance, has a lot of species have the same combination of the traits, and this happened in the Pacific is very clear between when you compare the Pacific between another different zones. But in the Atlantic, this maintain the pattern, but you can see here the difference between upper and lower is not very different compared to another areas, for example, for the shallow and the upper. In the opposite way, we see it when we start to explore the functional specialization, how different is one organism when combining the traits, can to see the opposite pattern. In other words, can to see the Atlantic in general in all the different levels, and when it start to increase the deep, it start to see more rare species in comparison for the Pacific. In other words, all the initial prediction for Rocha and collaborators, we say the community is totally different with can see in functional terms. This is a couple examples I have to show how can to use from the local to global scale the functional traits. And now I wanted to start to discuss how can you use the trait-based approach to understand the past and the future and to understand human impacts, for example. We probably all these people in this room remember a couple of years ago, lockdown during the COVID time, and many different papers coming to say it. The many organisms never we see before, they start to see again because dawn has more human impacts. And this was positive in many different ways for different organisms. However, for marine organisms, this increased the illegal fishing in different marine protected areas. This is one example here. This is one specific island here in this and the Brazil. This is the nail Alcatraces. And during the lockdown, when the police don't come to surround about the marine protected area, many people go for these areas to try to fish in there and increase the capture of the different animals there. When we start to explore what happened in functional terms, we start to compare, for example, for professional boats is more an amateur boat, is more recreative people. Basically, can sit here in yellow. My functional space for professional boat is cute in comparison for the amateur boats. But when I start to explore in during the COVID time, see how much increased in my functional space with amateur boats. In other words, the people start to go more there and start to capture more animals with high values and very endangered species. For example, you can see here a lot of charts start to include in this moment. In other words, I can use my functional approach to try to mapping what are the consequences when you have these another animals here. Another good example was this paper was published a couple 2 years ago, we basically compiled all the information about the fishing arts in Brazil. You can see the difference between the north for the south, the many people using all different arts here and the south using this specific art. I had one specific targets there. In the last 15 years, the people, you can see the core hit, how much the total landing start to increase more for the artisanal is more strong in Brazil in comparison for the industrial. However, when it start to go more deep and related with how change in terms of the all this the north for the south, can see for example for the north is more artisanal in general, but in the south has more industrial boats fishing there. Okay, but this is in terms of the how many total land in there and the biomass, and don't include yet the traits. But when you start to include the traits there, for example, the diet, we start to investigate how the north, for example, start to fishing and change the profile of the organisms start to fishing there. For example, in the north, for the last years, the people start to fish and fish more sharks there, and basically to reduce all this proportion of the rare species with large size, top predators, and this profile changes across the regions. Basically, in the south, the charge is no more possible to see and it start to decrease a lot, but in the same time, it start to explore another type of the organism, move, in another words, for another direction. To simplify it, all this figure basically have all the combination of the traits here, all the species here, and can see how much change across the time the target of the species and the proportion of the total landing was developed in Brazil. When comparing in general for the functional terms, we can see, for example, all the fishing in Brazil is more focused in general for trophy, high trophy levels, and has one small proportion of the low levels here. I want you remember this figure to try to understand this another figure here. This is my time. Have here the first moment and separate for the seconds period of the time. But important to detail here, for example, for the north, the fishing captured more focus in these high levels here. But when it start to increase the time, basically the people start to explore another different species to maintain the capture. This is for the north, but start to see what happened here in the northwest, northeast. It basically can't to change, but basically leave high up to, trophy level to move for low level. Basically, the fishing capture start to expand. Has one expansion of my functional space and how it can affect my community. After for this work, was generate one specific document and suggestion to try to prevent or help this expansion of the functional space. In other words, we have different traits. We can use it for try to understand a system and a species, try to understand the respond environmental factors, can to revelate ecosystem functions. And this is important to try to integrate all these metrics in and using the functional trait to try to understand what happened there. I wanted to say thank you for all my collaborators. There's a lot of people involved in this research and the people from my lab. I started 2 years ago now, and all these people working and continue to working and produce more knowledge assignments related with the species on marine habitats in general. And I'm happy to receive any question you have. Thank you.
Thank you, Juan Pablo. That was great. We have some questions already, so I will just read them off since we have 10 minutes. So the first question comes from Marina Alves Mega de Andrade. Hi, Juan. Nice presentation. When working with multi-species functional spaces that span different trophic levels, such as mammals, fishes, corals, and other taxa, how do we decide which functional traits are most relevant? For a single group, like fishes, there are well-established trait sets. But from a whole community perspective, how should we select the more relevant traits to work with?
This is big question in general because it's not simple to decide the trait you will use. The first things is how you can relate the traits, for example, one specific category. For example, in all the studies I show, I try to decide the traits between growing, survivor, and reproduction, for example. This is the first stage. But when you do comparison cross taxa, it's more complicated because it's hard to find traits you can compare between the taxas. But the first moment is to start to see, hey, maybe I can use the body size. The body size is very standard trait I can to start to include there. I can start to include the diet, for example, very general description of the diet to try to include there. But after to do these two different steps, the next step is to do one sensitivity analysis and to see how much I can to you understand about the traits I use and see what is more appropriate to continue the analysis. It's long process, it's not simple, but this is one way to try to incorporate this challenger.
Great, thank you, Juan. The next question is from an anonymous attendee. What methods did you use to define beta diversity and species richness? What measures and what indicators? Did you use eDNA or videos, or did you dive yourself?
Okay, I think it's two different things here. First is the method to collecting the information there. I use it more in the water visual sciences in general, and now I start using video plots to try to understand the fish community there. And after the second part is to for calculate the beta diversity, basically a couple of years ago, Baselga and collaborators published one paper to say the beta diversity can to decompose to different components. You have the nestedness component and you have the turnover. Basically, you have high nestedness is basically your community is very similar when you compare pair comparisons, but when you have high turnover is basically between the point A, the community A to community B is totally different. And it depends. And the people is now start to expand this concept for the beta diversity for functional and phylogenetic. For the functional, we use the traits, for example, to calculate the distance. And for the phylogenetic, the people use the distance in the phylogenetic tree, for example, to see what are the difference there.
And for species richness, was it just the number, the species identities?
Yes, exactly.
Yeah. Okay, great. Thanks. Mark Lomolino asks, to what degree and how rapidly do you predict introduced non-native species to invade and converge on the functional space of now extinct native species?
This is big and good question. We know many different ways to understand the invasive species. For example, in Brazil, the one specific coral arrived there and he taken all the Brazilian coast. But to see this species really change the functional space, we don't see yet this event. Is for example, if you compare the terrestrial organisms, a couple of studies compare the extinction for the birds in islands and the current community there is more simple to understand. But for marine, it's very difficult. For example, for fish, the only record the extinction for rich fish community in one place is from Galapagos. The last is one plantigrade species. He disappeared, I think, 20 years ago, and never more the people see it. but the effect in general in the functional space is not relevant for yet. I think the people need to start to move and try to using all the terrestrial advance to go for the marine habitats.
Mhm. That's a good point. So we don't even know if species went extinct and which species
Yeah.
went extinct in the marine environments.
Yeah, I think it's depend on the group. If you go for mammals, is the record of the extinction is better. You know, Catarina Pimiento show in the first paper I show here how change the functional space with the extinction and the current species there, and how can affect the composition there. Has a couple words more for with charts, for example, to try to simulate what happened in this term.
Yeah. Okay. Another question from Iram Abbas, who's a PhD student. His question or her question is looking at the past to future arrow on the slide. If industrial fishing continues on this trajectory without major intervention, what does your biogeographic modeling predict for the structure of Brazilian fish assemblages by 2050?
If we continue in the same way, we all the time we move to try to and capture more and more different species to try to maintain the fishing level. The problem is the organism is no it's not possible to maintain this fishing procedure. You need to control different ways. And when one action is to create marine protected area, but I show one example during the COVID time, many people go there and yeah, it's not simple. It's many conflict interests in the same time. For example, it's impossible to say, hey, let's go to invest in this specific area, but need more studies. It's not simple. I think, especially for this scale, it's hard. I think the people start to move and have different reference to show this one. Is the people use, for example, the interviews to see what is the perception between the people in the past. For example, if you ask my grandfather, he remembered fish in these sites. But if you ask for the grandson today, this is the big site. You lost the perception of the animals, and basically for you it's more normal to find this fish. For my grandfather, is this one. In other words, it's hard because the human has short memory.
Yeah. So if it continues on the current trajectory, the fish will be this size.
Yeah.
Probably. Okay. One last question. We have a couple more minutes from Maisha Granolati Fernandez. Hi, Juan. Thank you very much for your talk. I recently came across discrepancies between authors regarding the classification of trophic groups between oceans. It is in one ocean, a species is classified with one trophic group, but in another ocean, it is classified in another trophic group. In global scale studies, how can we deal with discrepancies between authors from different regions regarding functional traits, which are likely due to intraspecific niche differentiation?
Yeah, this is another big challenge to try to put. The first, a couple of years ago, many people meeting together and describe, okay, let's go to using these seven specific diets, but this is very general, and this is the problem. For fish in general, it's very qualitative. It's not related with quantitative. Now the people start to do more and more quantitative classification. Is, for example, for birds, you have very good classification in terms of the diet, how much is the percent of the fruit and how much percent of the mammals is the animal can to consume. And is probably is better in comparison for the only qualitative data. I think so the next step to try to put quantitative weights to compare the diet between all the organisms across global scale. But in the middle time, we try to adapt and reduce the number of the categories, especially for global scale. When you go for the global scale, you go, for example, the Eastern tropical Pacific, we using only seven traits, or only seven levels of the diet. But as you go for the Indo-Pacific, where the people have more knowledge about, have more diversity there, for example, for herbivorous fish, have different levels, not necessarily the same functions or the same type of the diet you can find in the Eastern tropical Pacific, and this make it more difficult to do this comparison.
Mm-hmm. Thank you, Juan Pablo. We are out of time. We have to let Dr. Quimbayo go. I'm sure he's very busy with his lab and his work. So thank you again very much, Juan Pablo, for joining us. It was a wonderful seminar. And thank you, everyone in the audience. I'm sorry if I didn't get to your questions. Thank you very much. And please keep coming. Come see us and listen to the next funk lecture with Katrin Graham. It will be a great one as well, I'm sure. So thank you. Until next time. Bye-bye, everyone.
Bye.